Hello everyone, my name is Maab. I am a senior at Central High School, and today is my pleasure to present my MSAP 2020 project. This year I was looking at the data analysis of the changes in mRNA expression in taste cells. So what does that mean? Well, basically the goal in this study was to determine the genes that are involved in the increased response to glucose in taste cells, and we did this by looking at mRNA expression. DNA produces RNA, and RNA is what produces protein, but RNA is much easier to measure than protein. So in this study, we were looking at mRNA expression in order to gain a better understanding of the genes that are involved in the increased response to glucose in taste cells. And the taste cells that we were looking at were treated with a growth factor, and as a response to their treatment with the growth factor, became more responsive to sweet stimuli. And we know that they were more responsive to sweet stimuli because these cells went through single-cell calcium imaging. And what happens in calcium imaging is that you take your cells and you dye them with a fluorescent dye. This dye is known as Fura-2AM, and it's also a calcium-sensitive dye. So you take your cells, you dye them, and you give them a stimulus. In our case, the stimulus was glucose. And then you'll know if there are changes regarding the calcium on an intercellular level because of the binding that occurs with the Fura-2AM. So the computer will pick up on that and it will plot it. And in our case, after 5 to 7 days after our cells were treated with the growth factor, these are the results that we saw. On the x-axis is our stimulus, which is glucose, and on the y-axis is the percent responsiveness of the cells. The orange here are the cells that were not treated with the growth factor, and the gray are the cells that were treated with the growth factor. So we see that the cells that were treated with the growth factor, many more of them are responsive to glucose. And at this point, the question became why, and we tried to understand the why by looking at the gene expression. And so the first step really was to treat the cells to create our two groups, our cells that were treated with the growth factor and our cells that were not treated with the growth factor. And the calcium imaging assay showed us that the cells that were treated with the growth factor, many more of them were responsive to glucose. So at that point, to understand that, we performed a chip array assay. And what a chip array assay does is it tells you how much mRNA is in your sample. And in our case, the chip array assay was performed at the University of Pennsylvania, and it was performed on both groups of cells. The groups of cells that were treated with the growth factor and the groups of cells that were not treated with the growth factor. And so ensemble and string were used for data analysis purposes. And after 5 days of our treatment of the cells, we saw that there were significant changes in the mRNA between the two types of cells, and that indicated to us that there was something going on on the genetic level. And so that is where my role in the experiment came. I did a data analysis on the results of the chip array assay. And so we had over 65,000 gene transcripts. And so I set the criteria of the data that I would look at. I looked at the data which had a p-value of less than 0.05 and at least a twofold change. And in these next few slides, you're going to hear me use the terms upregulated and downregulated a lot. And so basically, the downregulation in terms of a gene refers to a gene producing less of a cellular component. In our case, the cellular component that we're talking about is RNA. So when I say this gene is downregulated, I am saying that the cell is producing less of this cellular component. And when I say this gene is upregulated, I am saying the cell is producing more of this cellular component. And so you'll be able to tell how much more or how much less by looking at the fold change. And on the screen right now, what you see are the genes that were downregulated by the treatment. And again, they all meet the criteria of having a p-value of less than 0.05 and a twofold change of at least 2. And so this is the network of interaction of these downregulated genes. And it's important to note that in our study, we looked at IRS2, GPR133, CPE, SLC27A6, and PLD1. And what's interesting is that of the genes that we looked at in our study, a lot of them, these three do not have an interaction. So what that could mean is that these cells have their own role and they function independently. But it's also interesting to see that there is an interaction between two of our candidate genes, the IRS2 and the PLD1, but it's an indirect interaction. So that could mean that these genes do communicate with each other, but they do it via other genes. And now these are the genes that were upregulated by the treatment. Again, they met the same criteria of having a p-value of at least 0.05 and a twofold change of at least 2. And so again, we see that a lot of these genes are out here alone, so they seem as though they don't have an interaction, and that could mean that they possibly have their own role and they work independently. But we also see this time we see a direct interaction between two genes of interest, between the TMEM69 and the SLC30A6. So we see this direct interaction, and what this could mean is that these genes are connected, and that these genes may even influence the upregulation of one another. And then we also see a lot of indirect interactions, and again, that could mean that these genes are simply simply need other genes to communicate. So all in all, we saw that the downregulated genes mostly had an indirect interaction with each other, and we saw that the SLC, the SLC27A6, the GPR133, and the CPE have no interactions. But it's important to note for both the upregulated and the downregulated genes that just because this analysis doesn't show that an interaction exists doesn't mean that there are not any interactions that exist. It's possible that there are interactions through adapter genes or connected genes. And if this experiment were to be performed again, I think it's very important to reevaluate the criteria set for the data that would be evaluated. I think it's important to consider changing the p-value or the fold change, and specifically the p-value, because just because a gene is not statistically significant doesn't mean it doesn't have a direct interaction with one of our genes of interest. So I definitely think that's something that's very important to note in the future. And with that, I'd like to give a special thanks to the Monell Center and to Katie for being the coordinator of this program. I'd also like to give a special thank you to the Christopher Ludwig Foundation for supporting this research. And I'd like to give a special thank you to my mentor, Dr. Osdenver, who definitely spent a lot of time walking me through this research, and to Dr. Malin and Dr. Brunson, who are the muscle that keep this program running. So thank you.